Hey, 8th grade, Mr. Locker and Mr. Hill here to walk you through the meiosis crossover activity. So what you'll need is you'll need a handout, 2 pieces of paper that look like this, that include a total of 4 chromosomes. You'll note that each chromosome is in its copied state, which is why you'll see 2 copies of each chromosome. You'll need some scissors and some tape. As usual, begin by reading through the whole background. That will give you a review of some information that you should already know, as well as a little bit of new information, particularly about diploid and haploid cells. When you get to the second page, this is when you're going to be following the procedure step by step. Get 4 chromosomes. 2 chromosomes are blue. 2 chromosomes are blue. 2 chromosomes are red. The blue ones are from the father. The red ones are from the mother. You'll begin by cutting out the chromosomes. Save you some time. I've already done that. Remember that during interphase, each chromosome was copied, which is why at this point we're going to have 2 copies of each of the 4 chromosomes. So we're going to begin by taping them together to create something that's kind of like an X. To be honest, here it's going to look a little bit more like an H. So I'm going to tape them together here, like this.
Actually, make an X. It'll work better for
Right,
crossover.
it'll work better.
Make an X, yeah. There you go. Perfect. Look at that. We probably should have the tape ready, huh?
Like the pieces of tape?
Yeah. Do you make those sound effects in class too, Mr. Locker? I do.
They just happen naturally.
Yeah, me too. I do the exact same thing. Our kids are gonna laugh at this. I'm like, whoopsh, whoopsh. All right, let's see these X's.
Okay. So what we have here are 4 chromosomes, or 2 pairs of what we would call homologous chromosomes, and they're in their duplicated state, which is why you see them as X's as opposed to single lines. Okay, next we're going to go on to 3. Place your homologous chromosomes on top of each other to simulate crossover that occurs during prophase 1. So I'm going to place it like this. And like this. Kind of feeling like, there we go. And then you are going to somewhat randomly choose a point to cut 2 spots on each of the chromosomes, and that's going to be the location of the crossing over. You're going to make sure to not cut at the centromere or through any of the letters. Each letter is meant to signify a gene. So I might start by cutting here. And I'm going to switch. So this is where genes from one of the homologous chromosomes has switched or crossed over to the other. I'm going to tape them together. And another crossover event here. Tape them together. Same thing over here. Bear with us. We'll be done soon enough. And maybe we'll go here.
So just 2 per
Yeah.
chromosome, right?
Yeah, I should have maybe gone on the other arms, but it's okay.
That's okay.
Try to go fast for you so you guys don't have to spend 10 minutes watching me cut and tape.
All right, now let's get these guys closer.
So now I have completed crossover, and this would have really happened in the prophase 1 phase. And at this point, now I'm going to be on to question 5. I'm going to complete meiosis, and I can use my notes, more information up on the whiteboards, and then I'm going to record the genotypes here. Should we go through the whole meiosis? We should. So, you can imagine that this is my cell here. So my homologous pairs are going to line up at the center of the cell. This would be metaphase 1. Anaphase 1, the homologous pairs are separated to either side. Telophase 1, 2 daughter nuclei would begin to form. Cytokinesis 1, 2 new cells are formed, so the cytoplasm and the cell membrane would divide. I would have a stage where the DNA decondenses, but we can't really show that here. So next we're going to go to prophase 2, where the DNA would recondense to form chromosomes. There is no additional crossover event. Crossover only occurs in prophase 1. Metaphase 2. Chromosomes will line up in a single file line down the center of the cell. Anaphase, so I actually have to cut them. Chromosomes will be split in half. And just a reminder that it will be the spindle fibers that will be moving the chromosomes throughout the cell. Oh no. Luckily I can retape it. Don't worry, we don't need to start over. That would be a bad thing for the cell though. So again, we are in anaphase 2, when the chromosomes are pulled in half. These separate. Oh, another split.
This might happen to you. Just get some more tape in, fix it.
So these get pulled to either side, and then ultimately what we end up with are 4. Each of these would be the chromosomes in each of the 4 daughter cells. And as you can easily just see by looking at it, and we'll just do kind of a more sophisticated analysis, each one is different. So each daughter cell is genetically unique. Last thing you'll do is you will say this is going to be my gamete number 1, or daughter cell number 1, and you're going to record the genotype of that gamete. So we're going to have little a, little b, big D, and I'm just listing it down here, little e, and so forth. You'll do that for all of the alleles on each of the 4 gametes or daughter cells. Anything else to add? Well, I suppose we will continue with this, but that's what you want to get through with for today.
Thanks, Mr. Locker. I don't have much of a voice today.